Hello and welcome to Science Research Weekly, Season 4, Episode 14, Biologists Mapping Stuff. Get ready for majestic herds of prehistoric rhinos, open source 3D printed robot parts, a mitochondrial disease therapy, a global map of human subcellular architecture, potential but scanty microbial life on Titan, and oxygen adaptation in bacteria. This episode covers materials from April 4th to April 10th. Let's get started with some research articles. First up, enamel, carbon, oxygen, and strontium isotopes reveal limited mobility in an extinct rhinoceros at Ashfall Fossil Beds, Nebraska, USA. The Ashfall Fossil Beds preserved 20-million-year-old animals, and you guessed it, volcanic ash. This includes over 100 ancient rhinoceroses, Teloceros major. But the question is, was this evidence of herding behavior, all these rhinoceros together because they're a herd, or simply refuge-seeking behavior prior to the ash? To answer this, researchers sampled molars from 13 individual rhino fossils and checked isotope ratios. The results suggest that they were local to the Ashfall area and were limited in dispersal, meaning they did appear to form large herds. Moving along, we have the article, Development of an Open Source 3D Printed Material Synthesis Robot Fluid, Hardware and Software Blueprints for Accessible Automation in Materials Science. Fluid, so Flowing Liquid Utilizing Interaction Device, is an open source 3D printed robotics platform that looks to be a good way for a variety of projects that involve precision movement of fluids. Now, the article itself is behind a paywall, so that precludes deeper exploration of what actually is going into it, but there is a short video on YouTube, link in the description below, that shows it in action. If you are looking at the video portion of this, you can see that it kind of set up and precisely measuring liquids from plungers. All right, next article, Small Molecules Restore Mutant Mitochondrial DNA Polymerase Activity. Mutations in the gene, POLG, P-O-L-G, result in severe progressive conditions with high mortality and morbidity in humans. Because POLG is important for mitochondrial synthesis, so if there's a depletion of mitochondrial DNA, this really causes all sorts of havoc because cells need mitochondria for energy, and these sort of diseases in humans have no cure. However, researchers found that PZLA, a small molecule, can actually restore function in many of these POLG mutants, which means it's a promising therapeutic to mitochondrial diseases. So really interesting stuff there. Moving along, we have the article Multimodal Cell Maps as a Foundation for Structural and Functional Genomics. So researchers create a global map of human subcellular architecture, combining, you can see here if you're looking at the video, figure 1, combining things with like interacting proteins with antibodies, sort of that sort of data in these cells, as well as immunofluorescent images, putting together using machine learning, all sorts of things, coming up with these really cool architectures. And the thing about it is, of course, in any good article like this, there is going to be a link to the actual website that has these data on it. Here we go. We can look at the cell map. And looking at this multi-scale integrated cell map, you can look at the tree view. You can also look at the cell view. Of course, you can zoom in with the various components. There we go. All right, we will leave that for now and move on to our next article, The Viability of Glycine Fermentation in Titan's Subsurface Ocean. Researchers suggest that while microbes utilizing glycine fermentation as a metabolic pathway could be possible on Titan, Saturn's largest moon, glycine availability is limited by temperature, derived as such by impact melt pools. So the total number of microbes in this hypothetical biosphere would be very low. So they could utilize glycine, but there's not a lot of glycine, so there couldn't be a lot of them. All right, last up here, we have a geological timescale for bacterial evolution and oxygen adaptation. Researchers constructed phylogenetic trees of bacteria in deep time, especially looking at oxygen adaptation, the switch from anaerobic to aerobic. The last bacterial common ancestor likely lived 3.9 to 4.4 billion years ago. And the researchers found actually 3 bacterial lineages that switched to an aerobic, so oxygen-using, state prior to what's called the Great Oxidation Event, which is 2.33 to 2.43 billion years ago. So those earliest aerobic bacteria actually emerged 90 million years before that Great Oxidation Event, so almost a billion years before that. So oxygen use predates that Great Oxidation Event, where much more the modern lineages that use oxygen originated or switched to oxygen. All right, with that, let's turn to some research tool. There are 4 new CRAN packages. Correlation R, conduct robust correlations on non-normal data. SignFit R, modeling the relationship between nematode densities and plant growth. DLMRMV, distributed linear regression models with response missing variables, and LME4GS, LME for genomic selection. That's all I have for research tools and, in fact, research funding, because there's no new. So to kind of round things out and have something more than just a bit of a bust at the end, I have a question for you. Are you interested in an overview of clinical and translational science? If you are, you can check out the newest didactic training from the research group I'm part of, Transcend. The Professional Development Corps has different didactic training. The first one was an overview of clinical and translational research by Dr. Stephen Powell. It's a really great overview with some phenomenal examples. So if that's in any way your wheelhouse, I'd suggest you check it out. Aerobic Cherobic, thanks for joining me for Science Research Weekly. Science on.